It's so weird that two died when I go out.
I saw it.
I've only been to the water. I just go there and she takes me. They do. I know they do this shit.
Oh, yeah.
Big Island's got some good there.
Oh, yeah. I go with my buddy Glenn from Scotland.
Mary Hagedorn, yeah? Everyone can hear me right now? Hello? Yes? Okay, I would just raise my voice then a little bit more. So, I would like to introduce Mary Hagedorn. She received her PhD in marine biology from Scripps Institution of Oceanography and has been a research scientist at the Smithsonian Institution for 17 years. She worked throughout the world, from the Amazon to Africa, and in her current research she uses cryobiology for conservation of fish and endangered coral species. Please welcome Mary Hagedorn and take it away.
Okay.
Bring your mic away.
Can everyone hear? That's a little loud. Okay. Can everyone hear me? It's still a little loud. It's driving me crazy. Yeah? A little loud.
For us, it's good.
Okay. All right. Okay. It's a pleasure to be here, and I'm delighted to be talking to you today, and I'm going to try and tell you a little bit about the conservation work I've been doing for the past 10 years around the world, but mostly in Hawaii. I have many collaborators. I just put some of them on this slide, but I'll try and show you some of them. But let me just give you the big picture first. So if you want to sit and drink and phase out a little bit, you can. So I'm going to tell you about coral reefs. They're some of the oldest ecosystems on our planet, okay? And they are in trouble. And we are in trouble because they are in trouble, quite frankly. So I'm going to try and give you the message, or leave you with the message that coral help us live and why they help us live. So here are some of my collaborators, Mike Henley and Jen Carter. Jen is my assistant at the Smithsonian. Madeline Van Oppen at Ames, which is the Australian Institute of Marine Science, and Rebecca Spindler at the Taronga Zoo in Australia. But first, let me start by saying thank you to Sea&Learn. It's a great pleasure to be here, and I'm very happy to be here. And also Paul Chisak and the Shearwater Resort for hosting us. It's a beautiful place. If you haven't been, please go. And to Brigadoon, to Michael and Trish for hosting it tonight. Thank you so very much. And then I have a host of other foundations that help me. But I am a huge Star Trek fan. I was as a child, and as I grew to be an adult, I wasn't a Trekkie exactly, but I always liked sci-fi and I liked Star Trek. And then one day, Star Trek called me on the phone, and I thought it was a joke actually. But they actually have a foundation. It's the Roddenberry Foundation, and they're now supporting some of my work, which is fantastic. But I wanted to start out with pointing out the latest movie called Into Darkness. And the reason I want to talk to you about it is because they use cryopreservation in that movie. And so, I don't know if any of you have seen the movie. All right, one person, great. So, two people. In the movie, there is a very smart bad man. And apparently he showed up in the TV series in the 70s, and his name is Khan. And he's genetically engineered, and his whole population, he has a group of people who are genetically engineered, but they're bad guys. And so they stick them away and they cryopreserve them. But in this movie, Into Darkness, someone lets him out, they revive him. And it's all about, sort of how he wants to revive the rest of his population. But they talk about cryopreservation and the need to sort of keep things for the future. So that's really what I'm going to be talking to you today about. So I'm from the Smithsonian. This is the castle, one of our very first buildings at the Smithsonian. It's 150 years old or older, really, now 170. It's the largest museum complex in the world. And I work in the Department of Reproductive Sciences, and most of my colleagues work on very charismatic megafauna, pandas, cheetahs. It's very hard to have coral have any traction when you have all these extraordinary organisms that people are very interested in. I mean, one woman threw herself in front of the plane at Dulles Airport to stop the panda from going back to China. So that's true devotion for you. But one of the things that we all have in common is that we all deal in some way with maintaining germplasm, that's sperm or eggs or embryos, frozen in some state for these animals. So that's what the doer here represents, is that we cryopreserve in some way these organisms. Okay, so why do we want to do this? Why is it important to try and make banks? Why do we want to do this? And this is probably one of the best examples in the world today of why it's important. Now these cute little critters here are black-footed ferrets. They were considered extinct, I'm going to say in the 80s. And then a dog found one on the prairies of Wyoming and brought it into a farmer. And the farmer, of course, knew what it was, because there was a concerted effort to kill the black-footed ferret because they ate prairie dogs. And prairie dogs make holes in the ground, and the farmers and the ranchers hated the prairie dogs. And so we have these pictures from the 30s of mountains of prairie dogs that had been poisoned. And as the prairie dogs died, then the black-footed ferrets died. And so it was amazing that they found these black-footed ferrets, and there were 100 of them in the population at the time. And so, of course, we're the government. We argued, and we argued, and we argued. And during that time, distemper went through the population because the dog obviously had some kind of distemper or something, and 80 of them died. So now we're left with 20. And they brought them into captivity, and then they started the most extensive dating service you've ever seen. Each prairie dog had its genome done, and they mixed and matched them. But the most amazing thing is my colleague Jo Gail Howard froze the sperm from these prairie dogs. And so even the individuals that were in the population back in the 80s are still part of the breeding population today. And we have over 4,000 of them back out on the northern plains. So it's an example of using cryopreservation to expand populations and put animals back into the wild. But the thing about having a bank, a frozen bank of any type, whether it's cells, anything, is you can look at the history of disease and epidemics. This is really important for us to understand. It stops evolution, which can be a good thing or not, but you can also use them to help diversify the populations, which is what Jo Gail did with the prairie dogs. She used this bank consistently. But the other thing that's extraordinary about these banks is they give you perfect, absolutely perfect genomics. And a lot of people are talking about genomics today and how we build, and look at genomics. And the cells are intact, and they're good for at least hundreds of years that we know about. So the cells are frozen but alive. And last but not least, it can prevent extinctions if we use them wisely. Okay, so on to coral. This bubbly thing kind of is a synthesis of what we're talking about here. So let's start out with the benefits of coral reefs. They help support our economy by fishing, and any island that has a coral reef, almost 80% of that economy is based in some way upon a reef. I'm sure this island is no different. They provide nurseries for fish. They're coastal buffers. They produce, coral reefs don't, but they help produce oxygen and are involved in the carbon cycle in our oceans. And they can help produce new drugs. And certainly they are a source of food because of the fish that live in them. Now, in terms of the threats that are facing coral reefs, we have acidification of our oceans, pollution, sediment, warming temperatures, overdevelopment, overfishing, and disease. Going to go into all of these in some detail. Okay, so we live on an ocean planet. 70% of our world is the ocean. And like the trees in the rainforest, our oceans produce most of our oxygen. Prince Charles got it wrong. He said the rainforests are the lungs of our planet. It's actually the ocean that are the lungs of our planet. 50 or more % are produced in our oceans. But the coral reefs are the jewels of our oceans in that they host so many individuals but they also are critical in how we manage our carbon on our planet. And as we know, we all have carbon footprints now, and we have carbon this and carbon that. The coral reefs are really instrumental in helping us in the oceans with managing that carbon. So I heard this thing that they're small. They only comprise about 0.2% of the Earth's surface. So I said, okay, what country is about that size? And it's about Bolivia. So all the coral reefs in the world take up about this size, which is pretty small when you think about it. They are eco-dynamos. And as I told you, they provide nurseries and homes. And we did a great little thing with the kids today down. These guys helped us. We did this whole thing with the reef as home. Because really, reefs are apartment complexes, and they're living structures because they are animals, but they provide homes for all these animals. And we know when reefs disappear, your fish disappear. Because there's no more homes, there's no more safety, there's no more ways to get away from predators, etc. They also perform natural buffers against storms. And living in Hawaii and having experienced 4 tsunamis in the last few years, we have a barrier reef in our bay, and I am very thankful that we have it because our home was not impacted by any of the tsunamis. And they are also a source of potential medicines such as antibiotics, cancer, and AIDS. NIH is doing a really good job of collecting coral and looking at some of the marine byproducts for drugs. And as I said, millions depend upon them for their livelihoods, such as fishing and tourism. Okay. Great Barrier Reef brings in $6 billion annually because of their reef and their fish. It is suggested that reefs bring in about $300 billion annually across the globe. No one really knows what the actual number is, but it's what they suggest. All right, so here are some of the reasons that coral are having difficulties around the planet. They're both local and global. Locally, things like sedimentation. This is a satellite view of a river. You can see the sediment just pouring out here. It smothers the reefs. It basically covers the coral, and if they don't survive, the coral recruits cannot burrow down through the sediment to survive. So it quickly destroys reefs. So there's many things like this. There's pollution, all these things pouring out of our rivers, too many nutrients, etc. And then overfishing. We have some very destructive fishing practices, from dragon trawls to dynamite fishing. Even just overfishing is a horrible thing. In our bay, again, Kaneohe Bay, there is no limit on fishing. You can take as many fish out as you want. And when you go to look at the reefs, there's these tiny little fish and nothing else. And coral reefs and fish live in a community. They are symbiotic, and you need to have fish and a reef in order for it to be healthy. So overfishing is a really bad thing. So now here are some of the global causes. We are basically the global causes. And we are overusing fossil fuels, and we're putting fossil fuels into CO2 into our atmosphere. It's blanketing the earth and causing the air to warm and our oceans to warm. Most coral have lived at the same temperature for hundreds of thousands of years, just a range from, say, 23 to 30 degrees centigrade. And they haven't varied for hundreds of thousands of years, and now we're boosting them to 32 or 33 degrees. You had a warming event here in the Caribbean around 2005. You lost a lot of your coral in the Caribbean, and particularly here in Saba. And it's because of this type of warming. The warming, and in addition, think about the ocean as a sink, and as you're putting CO2 in the environment, it's sucking down CO2, okay? So it's holding a lot of CO2 that we are putting in the air. And so as we're putting CO2 in the water, it's becoming more acid, just like this soda is acid. It's the same process, basically. Mother said, don't drink so much Coke, right? She said that. It's not because of the sugar in it. It's okay if I use this as a prop. A lot of it is because of the acid. It can erode your teeth. And our teeth are very much like the coral. And so the acidification of our oceans is going to cause great harm. So this complex local and global things are causing stress. Coral are animals. We are animals. It's causing stress, and they're getting diseases. And if I had to say what's going to cause the greatest harm to coral in the world, it is disease. More than, I mean, these are the ultimate factors, but disease moves so fast, it's hard to even manage. Okay, so here are some diseases, and some of these diseases we do not even know the name of, and they can rampage through a population in just weeks. Here's a beautiful, okay, you should see this outside your doorstep, and I'm very sad that you're not seeing these outside your doorstep. This is a Cropper palmata. It was the foundation for reefs in the Caribbean, as big as a VW bus, most of them. I mean, they're just fantastically beautiful and just gorgeous, gorgeous animals. And here's what a healthy Cropper palmata should look like. In weeks, you can see it's completely decimated by disease, and this is massive, massive animals, just reduced in weeks. And now I just want to show you kind of how some of the changes are going on in the reefs. So here's the Bahamas Rainbow Reef in 1991. It looks pretty nice, got some nice coral here and sand and stuff like that. 10 years later, basically, it's just covered with algae and it's rubble, and unless you can really show people their reef and what's happening in the years that have happened, you don't track it. You think everything's fine, but seeing these pictures, you go, oh my God. And I certainly have that problem as well. This is Kaneohe Bay. It's right off where my lab is, and I go, ha, beautiful coral, a couple of fish, so nice, This is what it should look like. This is the northwestern Hawaiian Islands. It's not far away from us, but this is what a real Hawaiian reef should look like. Packed with fish, packed with predators. My friends who go diving there say they get in the water and see 200 sharks within minutes. I have never seen a shark in Hawaii. I mean, in a tank, but never in the water. I know they're there, but it's not like this at all. So we are changing our waters. So now in the Caribbean, we have a Cropper palmata and a Cropper silvercornis are on the endangered species list in the US. And what happens you can still find them there. They're still off of Saba, but they're there because the ecosystems are collapsing and the fish are starting to disappear. There's little or no coral breeding now in the Caribbean, and that's the biggest problem that I foresee, especially the species of Cropper silvercornis. We know they still breed in Belize, but we don't know any other place in the Caribbean where they're breeding. So that means there's no adaptation to changes, and there's a loss of genetic diversity. So this is a very bad place to be. And I just wanted to show you what a real, how big they can get and what they should look like off of Saba if we still had healthy ecosystems. And this is the one I was looking for here. This is the silvercornis population, and the captain of the Sea Dragon very proudly told me there was a 6 foot by 6 foot piece of this near the harbor, and I was like, oh my God, it should look like this everywhere, all over the island, and it should be covered with this. Okay, but so here's another possible solution. As we're losing species, maybe there's other species that will crop up and might be the salvation. And this is a hybrid. It's very interesting, and it's a hybrid between a Cropper palmata and a Cropper silvercornis, that's saccharine elkhorn, and it's called a Cropper prolifera. This is in Belize, and it lives in a really hot water. I mean, I told you that coral don't like to be heated too much. They start to stress, but these guys love it. They love hot water, and they love the surge zone, and they're doing really well in Belize. They weren't that prominent until about five years ago. We saw them, but now they're starting to breed. And so there may be some things that go on that allow natural selection to help us with this whole process. Okay, so now we're just going to do a thought experiment. What if coral reefs disappeared? The major thing, and I think IUCN agrees with me and many, sort of food organization, if reefs fail, hunger around the world will increase. There's no question about that. We are losing our fish already, but when we lose the nursery grounds, hunger is going to be a huge issue in terms of the world. And it has been suggested that 95% of our stony coral will be gone in 40 years. And who knows if that's really going to happen? I don't know the answer. So we asked the question, could we help? We can't stop these processes, but what we can do is we can create a bank, and we can say, in the future, if you want to diversify a small population or you want to receive the ocean, we can have that for you in the future. It won't be my future. it'll be grandchildren and great-grandchildren's future of the people in this room. So I want to just basically explain to you that coral are animals. That's the major take-home. Let's go back here. Major take-home message here. And here's what an individual polyp looks like. Okay, they kind of look like small anemones. And then, many of these polyps are genetically identical in a colony or a piece of coral. When they reproduce, some of them produce. So coral generally are hermaphrodites, both male and female. Some are sequential hermaphrodites, that they're a male 1 month and a female the next, or whatever. But they produce some type of gamete, either eggs or sperm, or egg-sperm bundles. And in this case, here are the eggs. And think of it as a cluster of grapes with a package of sperm kind of in the center. And so here's this white part is the sperm. And we collect these bundles, and then we kind of break them apart, and we collect the eggs and the sperm. So this next slide, I'm going to show you coral spawning. This is from Coral Sea Dreaming, which is just a beautiful, beautiful. So double click on that. And so you're seeing the egg sperm bundles being secreted, yeah? That's a good word. And I'm going to show you a couple of different species so you can see that it's different. So these are going to start to break apart. You'll start to see that eggs kind of, so you can see all the different eggs and how it's in a cluster. There, you can see that's breaking apart there, but there's tons of eggs in these guys. And the next one is going to be a mushroom coral, and it's going to come out like smoke, and that's sperm coming out into the water. And we actually have a hard time collecting this. We have to be right over them with pipettes. So we just try and catch it right like that. And this is a brain coral. It's all going to come out at once. See it coming out? Most of them are filled with lipids, and so they rise to the surface. Some are not. Some species are not. Okay. And so here in Hawaii, this is Kaneohe Bay, and this is this coconut island where I work. Kaneohe Bay is just a really natural laboratory for coral. It's one of the few places in the world where you can step out of your lab and get coral, like right here. And it has extraordinary resources on the island. We have a confocal microscope, we have radioactivity, we have all sorts of genetic, we have a very active genetic laboratory. We just have everything we need to do coral research there if we need to, or physiology. And we have patch reefs, so these small little things are patch reefs, and over here on the edge is an actual barrier reef. So we have all examples of reefs here. And we can either work in the bay or we can bring fragments in. And these are some collected fragments from Australia that are spawning these beautiful red, kind of looks like Christmas, these beautiful red eggs. But we can bring fragments in very easily into our lab, into our running seawater. And that's my assistant Ginny and I are here. This is mushroom coral. Most coral breed once a year. Okay, you're spawning once a year, and that's not a lot of time to do your work in. What's great about mushroom coral is that they produce eggs. This is sperm, okay, and this is eggs coming out here. So they do that 5 times a year. And it's usually all coral reproduces in some congruency with moon cycles. And for many species we know exactly, like if you come and want to work on mushroom coral, I can tell you the exact day and the exact time that you'll have to be out there. So it's sometime 2 days after the full moon at 5 o'clock. And every coral is this way. They only have about 10 cells, but they are synchronized extraordinarily. And so time of day, seasonality, and water temperature all come into this synchronicity for coral. And most, as I said, only breed once a year. So a lot of the really big coral populations, the Great Barrier Reef, that sort of thing, once a year. And it's this massive, hundreds of species are spawning at the same time. Here in Kaneohe Bay, we have about 8 species, and they breed. It's been disjuncted because Kaneohe Bay is so quiet. So many species will breed 3 or 4 months in a row, and they aren't breeding all at the same time, which is great for us. So there's asexual fragmentation, like taking a plant cutting. Okay, so coral can reproduce that way, but it's a clone. Okay, it's not. And then there's sexual reproduction, which is broadcast spawning of some sort. Okay. So this is our model that we've been using to try and develop. I when I first went out to Coconut Island, no one had worked on cryopreservation for coral. There's many species that cryopreservation has been successful with, but nothing on coral. They're very easy to collect. You can just snorkel and pick them up off the. This is only probably in 5 or 6 feet of water. So just briefly, as you have eggs and sperm that are being produced by the coral, they form a larvae, or an embryo, and then a swimming larvae, and then it starts to settle. And at some point, they get algae inside of them. So most worm coral have algae called zooxanthellae, and those feed the coral. They're symbiont. Okay, so here's Ginny. Usually what we do is we put the fragments, we go out before we know the coral's going to spawn, we collect from a number of different patch reefs, so we have genetic diversity, and we put the fragments in these floating for 2 purposes. One, for stem cells. We are freezing the stem cells of coral, and we'll also send some of this coral back to aquaria around the world. So we're doing captive breeding, we're doing captive rearing of the coral. And here's our first juvenile Acropora palmata ever reared in captivity. One of the things that I think is really important is that we're not taking advantage of our aquaria. We could take most of the coral that are endangered and start rearing pieces of them in aquaria, and then it would form a live bank. I'm forming the frozen bank. I would really like to see a live bank around the world for coral, because we could keep them for hundreds of years and just keep cutting them back like you would a plant. Okay, so what elements of the coral reefs can and cannot be cryopreserved? Coral eggs cannot be cryopreserved. Now, many of you probably have relatives that have had, some kind of assisted reproduction. There's over, I think, at this point, 2 million people in the U.S. who have been produced through some kind of artificial reproduction of some sort, whether it was artificial insemination or freezing of sperm or you name it. And until recently, I'd say the last 10 years or so, human eggs were not well cryopreserved. They would do it in cases of when women had cancer or something like that, but you didn't really get very good results. We have very new modern techniques where we use very fast freezing rates, and now almost 100% of women's eggs are revivable, and they have very good implantation rates. I just went to a whole human reproduction thing. It was amazing. So here's a coral larvae. You can see its little sort of hair, and they can swim around. But if you chill them, and this is true with the coral eggs, they just fall apart. So this does not allow you to cryopreserve them. And these are the algae inside the coral. These do not like to be cryopreserved either. They're very sensitive to cold temperatures. So we will probably end up drying these. I have some colleagues in Scotland who are going to help me try and dry these and use drying technology to get these. Because these are crucial, absolutely crucial. A coral has to have the right algae. If it doesn't have the right algae, they don't survive. And we know that the algae sometimes sets the tone. So the algae that live inside the coral in the Red Sea tolerate temperatures up to 40 degrees. Algae here probably only tolerate up to 30 degrees. So I have colleagues who want to engineer this and try and change some of the algae in some of the corals. Okay, what we can do is we can break apart these embryos. We can't do the embryos, but at this stage, the 8-cell stage, they're considered pluripotent. They can form into any cell in the coral, and we break them apart mechanically by just a kind of a glass mortar and pestle. And we get the cells, and we put the cells in these cryo vials. And now, this thing is called Mr. Frosty. It cost me $75, and I was so outraged that this cost me $75. And I could have gone to Target and bought this, pretty much. But it's an alcohol freezer, and actually you can do human stem cells in this, any kind of stem cell really. It freezes at about a degree per minute. You drop them down to minus 80 degrees, and then you put them into liquid nitrogen. We have very, very good recovery of these cells. And if we want to do them in the field where we don't have any energy, we can put them in dry ice, and that drops them perfectly to minus 80 for over 4 hours. And then we can put them into liquid nitrogen. Now, why is this important? Why do we need to cryopreserve stem cells? So we'll come back to human stem cells, and human cells could not be cultured until Henrietta Lacks. And Henrietta, I'm sure, if you haven't read this book, it's fantastic. It's a little plug for the book here. It's an amazing book about human medicine, and humans and how we get along. But she changed, her tumor changed medicine as we know it, because then human cells could be cultured. And right now we cannot culture coral cells. And we'd love to be able to do that because corals are dying of diseases and we cannot go out, and expose coral to healthy coral to various diseases. We'd love to be able to culture them. So if we can get our stem cells to grow in culture, then we can learn much more about coral diseases. And moreover, because they're pluripotent, that means they can grow in any cell in the body. We might be able to coax those cells into forming a coral in the future. So if that's the case, we have billions of stem cells frozen. Okay, what we can freeze as well is sperm, coral sperm, and they look a lot like human sperm. Okay, so this is our. I made this freezing method up because I wanted people around the world to be able to do this. I have very fancy equipment in my lab, but if I show someone in Indonesia, they're not going to have enough money and they're not going to be able to do it. I want to hand someone a styrofoam box and say, this is how you do it, and it's going to cost you $20 to do it. So this is the next model up. And this freezes perfectly at 20 degrees per minute. It just floats in the liquid nitrogen bath in this little styrofoam chest. And we get really good post-thaw motility. If you could just hit that. This has been frozen. Might have to hit it a couple times. There we go. So this is thawed coral sperm. And one vial we can use to fertilize hundreds of thousands of eggs. So it's one vial is very, very important. So just here's some of the sperm that we've cryopreserved from this species, which is a Cropper plumata, and fungi in Hawaii. And this is just really to show you that on each day, it's never the same. Even when we cryopreserve it using the same techniques and same everything, there's different physiology on different days, and we don't understand it. But we cryopreserve every night, and the stuff that doesn't look good once we thaw it, we thaw a sample out. We just have to prune our bank and we throw it out. Okay, so here's what it looks like when we're doing our in vitro fertilizations. So we have for smaller, to show that our sperm is working well and it can fertilize eggs. We have lots of small vials that we do this with, and lots of individuals that we do this with. But this is a much larger procedure here. This is in Australia, and we're doing millions of eggs here. And this is one of the results, our first result that we got. And this is Cropper plethrata, and I feel like this is my child, with baby pictures here. So this is produced from fresh sperm, and this is from cryopreserved sperm. And here you can see their little tentacles. They've glued themselves down, and these little brown spots means they've taken up their algae. Very important step. They've gone through symbiosis. So we know that the very first steps, the very crucial ones, they can fertilize, they can develop, they can settle, and they can form new, individuals. They can make their calcium, and they can take up their symbionts. But what we want to know is long term, is it going to make a difference? In humans, it doesn't. But people do not believe that this technology is useful for conservation, especially for coral, unless we can say, 3 years from now, this coral and this coral are exactly the same, and you were produced from cryopreserved sperm, and you were produced from fresh sperm, and now we can get you to, not you in particular, but we can get these individuals to breed. So that's what we're going to be doing, and there's a new facility. In December, we're going to be rearing millions of individuals in this facility in Australia. It's called Sea&Learn. Australians love cute names. Sea Simulator. And it can produce any characteristic of the ocean, past, present, or future. And we're going to rear these for 3 years, and then we're going to try and close the life cycle and get them to spawn in captivity. So that would be very exciting, because not only would it show that the bank is useful and that we have not damaged the coral by cryopreserving them, but it'll also give us a blueprint. So it'll say, here's your bank, and this is what you need to do in the future. See you later, kind of thing. So here's some of the species that we have in the bank: Palmata, Fungia, Acropora cervicornis, Acropora tenuis, and just some numbers, just so you would know. I mean, these are the 5 species we now have in the Australian bank. Oh, I forgot to put that one picture in. But they're billions, you can see, of sperm, and we have billions and millions of pluripotent embryonic cells. But unfortunately, this bank is sitting in a trailer in the middle of the desert of Australia. It's really not in a secure location at all. And so we're trying to now get the Australian government to consider forming a national bank. And so, unfortunately for us, the former director of the zoo, I work at the National Zoo at the Smithsonian, we had this very charismatic director who then became head of the Office of Public Management. And now he's become the new ambassador to Australia. So we're going to work the ambassador to see if we can try and get this very important material, along with other very important biomaterials like Tasmanian devils and things like that, that my colleagues have, frozen sperm for, into a secure bank. Okay, so here are some of the new developments. Now, you don't want to be just stuck with reproduction. It happens so, you have a few days a year to do this. So what we're trying to do now is we're actually trying to cryopreserve tiny little fragments of coral. And that means you're cryopreserving the algae, which is the brown part in this tips, any bacteria that's on the surface, plus the coral itself. So that's in process. And we're also trying to, now I was trying to explain this to some folks today, it's probably you. You have a lot of wonderful crustose coral and algae here in Saba. It's all that pink and red stuff that's on ledges and things like that. This is the most crucial player on the reef, believe it or not. It's very thin. It looks like somebody just forgot about it. But it gives off the cue that tells coral to settle. Without the information that this crustose coralline algae gives off, the coral really don't know how to settle well. So it's crucial, and it's very thin. It's made of calcium, and it's probably one of the things that are most affected by, or will be most affected by, acidification because it is so thin. We're desperately trying to get it preserved, but because it's hard to tell when it's alive even, we have to take pictures of it, and it's maddening it's very slow going. It takes us months to grow it, and then we take a picture of it. We're not sure it's alive or dead. No, it's really maddening. Okay. We're also working on reef fish because a reef is not just coral, it's about the fish. And this is a really wonderful little goby, and gobies really tell you, if you can look at the gobies, you know how the reef is doing, because gobies are kind of an indicator for the reef. And what's really amazing about fish is that you can take the testes of a fish, okay? So let's say you've taken the testes, right there. And you've taken the cells out and you've cryopreserved the testes. So you've taken the testes out, you've pulled the cells apart, and you've cryopreserved them. You can now inject them into another fish, the blue guys, they're not green guys. And these are sterile, so they don't have any gonads, they don't have any sperm, testes, or ovaries. You can microinject in these frozen testicular cells, and these blue fish will produce more of these green fish. So basically, just by freezing the testicular cells, and they're so easy to do, I can show anyone to do it in a heartbeat. And it's inexpensive. We're going to be banking these like crazy. And the Smithsonian, I'm really pushing the Smithsonian to make this their new way of collecting, not just dried things, not just things pickled, but that we would have a live bank that not only gives us the genomics of that species, but also allows us to create them in the future. Okay. So I've told you that coral sperm and stem cells are now in banks around the world. We have about 8 species, but there are 800 corals. So we have 1%, which isn't great. We're the only group right now doing this. We really need to start training more people. And we're also trying to cryopreserve and bank reef fish, their stem cells. And hopefully, we will never need them, but if we do, these cells can be maintained in the banks for hundreds of years. So how is all this interconnected, and what is the big picture in all of this? So here are these things called pteropods, and they're mixed in here with the sort of base of the food chain. So imagine, sort of all the plankton in the ocean. Pteropods are a favorite snack of whales and fish. Unfortunately, pteropods are being affected by acidification. They are dissolving. Okay, so their structures are not as robust as they were, say, 10 or 20 years ago. So if we somehow get rid of pteropods, we also may get rid of the whales and the fish that depend upon them. And certainly the bigger fish and the top predators will be affected by that. These guys produced our oxygen on the planet. They are crucial to our life on the planet. What is going to happen if something happens to those guys, and what will happen to our oxygen? We don't know the answer to that, but it is a very delicate balance that we do not want to affect. Okay, so I'm going to just leave you with this. I have one more slide after this. Recent studies suggested ocean acidification is progressing faster than we expected. I think in some species that's true, in other species it's not. But it's certainly not a straight line, and we don't know how it's going to affect them. So what can we do? Student, get a great education. Ask questions, try and reduce your carbon footprint. We're going to have a great solar talk coming up, and that'll be great to reduce the carbon footprint on Saba. Become active, whether you are a scientist or not. I think everyone has a voice, and you can help us train students, because the most important thing we can do now is start trying to spread this knowledge and get more people trained, because I can't do this alone. I cannot train, I can't be everywhere. So training and legacy building is really important. And I'll just leave you with this quote from E.O. Wilson that says, "Biodiversity is the planet's most valuable resource, and it's on loan to us from our children." Thank you very much. Questions?
How can you be so precise in the knowing when you're going to reproduce? I know that it's the full moon.
Yeah. So the question is, how can you be so precise? Really, it's not me. Coral have about 10 cells. Okay, so there's not many. We have hundreds of different cells in our body, but coral only have a few. And some of them have pigments in them that are similar to what's in our eye, the pigments in our eye. So that coral, as long as they are open to the full moon and the sun, they synchronize. And they're tracking the sun and the moon, and with the warm temperatures and things like that. We miss it. We're starting to miss it now because, for instance, I had an expedition to Belize to study there, and I know they spawn in August, the full moon. They spawned in July because it was so warm now, and it was so warm in Belize, so I missed it by a month. So we're starting to get these misses where we hardly ever did it before. But generally, once you know a species and you know its breeding habits, every year it's pretty much the same. And that's the other thing. In Hawaii, in Australia, all the coral breed. We're starting, breeding is a problem here in the Caribbean. We don't know if it always did or not. So there's a question in the corner.
How does cryo-freezing circumvent cellular lysis when you usually freeze something?
Okay, good question. He said, how does cryopreservation stop lysis? Okay, so I'm going to bring you back to your home refrigerator and your home freezer, okay? So everyone knows, and has probably done this experiment, we either have ice cream out or hamburger, and you decide you don't want to, you're in a hurry. So you just throw it back in the freezer, right? You don't put the top on the ice cream right, and you don't pack your hamburger or whatever up tightly. It'll start to get frost damage, right? And it tastes terrible when you eat it, and so that's ice damage, right? But, so I didn't put this slide in, but basically, here's a cell, and we use cryoprotectants, which are antifreeze solutions. And it's a very complicated series of sort of equations that you have to understand about different cells, how water comes out of a cell, and how these antifreezes or cryoprotectants can get in. Some are toxic, some are not. And so there's a lot of research that goes into understanding how different cells can get frozen down without cellular damage. But you walk a tightrope between damaging them and keeping them alive. And some are damaged, but some are alive. And so you do what's best, for the median and the population.
Do you just remove the water?
Most of the water. You can't remove all the water because proteins are held together. So, but you remove. They say 90% of the cellular water, and then there's different processes. Sometimes you can, instead of freezing, you can actually vitrify, which is similar to what is here. This is a glass. So that's what a lot of modern people a lot of modern techniques are using, vitrification. That's what they use on the human oocytes now, is vitrification and very ultra fast freezing, so no actual ice crystals form. It's a glass.
I have two questions, and on this, an analogy with plants, it's like an anti-desiccant, basically, sort of capturing
Let me repeat that question. He asked, it's like plants, it's like a desiccation of the process, and he's absolutely right. You want to get out the water, because it's the water that can make ice crystals, and they just pierce. It's like a balloon, you know, it can pierce through it. Once you've done that with your hamburger, your ice cream, it tastes terrible, the cells are disrupted. So yes, you're getting out the water as much as you can.
So the two questions. One, there are these experiments going on of using man-made materials or human-made materials as a framework for coral reefs, upon which the coral can then grow. But I hear that that's a bit controversial, or else they don't quite have that down right.
Okay. He asked, the question he asked was, what's happening with some of these man-made materials for encouraging coral to grow. And so let me be very clear. Every conservation method that we can come up with for coral reefs is important. I think we should not ever say, oh, that's a good one, that's a bad one. We need to do everything that we can. So some are working better than others, and I think some are, like for instance, here's a really good example. I was talking to Lynn earlier, and they've done some gardening where they've actually cut and placed small fragments out. But if you fragment coral in the summer, they stress and they die. If you fragment their physiology in the summer is completely different than their physiology in the winter because they're photosynthetic. And so you have a lot of lipids, you have a lot of other things. Everything changes because of the temperature and photosynthesis between the summer and the winter. So something like doing fragmentation in the winter is a much better idea. And so you can have these subtle things that people maybe not that aware of, and so things will get better. But it's not going to offset what's happening globally. So ultimately, it's important that we try to do these things for our populations. We should not give up hope. But I think ultimately, unless we can do a much better job reducing our climate change, we will lose our coral.
I have a second question, but I'll come back.
No, I was going to elaborate on your question. You said that artificial reefs were controversial. It has been done in Curacao, for example, where they did drop artificial reefs in the water. They're basically these domes with holes in them, and they're hollow, and fish can live in them, and coral can grow on it. The only reason they don't do it on Saba is because the marine park was set up to basically preserve what's there, but not to fix any damage or to add anything onto it, and that's why it simply isn't allowed yet here.
Did everyone hear that? Basically, some of the sort of auxiliary benefits of putting these reef balls, right? Are not done in Saba because it's a marine park and it isn't done yet here. So did you have another question?
The second question, you talk about whether it's Australia, the trailer in the desert, and trying to work with the government or getting more funding, and the struggle that you're having. Generally, when it is essentially the government support, it's a bit more publicly owned, so to speak. It's not. But in this race against time and the adverse effects, is there discussion going on about, for example, I think about Vinter Corporation, which is trolling the oceans right now and collecting biospecimens everywhere. But of course, they're going to have proprietary rights to everything they do. But they are very excited about trying and have money trying to do this. Now you sort of chuckled, so obviously I'm anticipating your response to that.
Okay, so.
But isn't that sort of, in this race against time, it means extinction or...
Yeah, no, I agree with you, but I feel very strongly that this needs to be maintained by the public. And, I've been approached by private corporations to do it, and I just said, you have to agree that it's going to be in the public, and then they back off. But your point about Craig Venter. Craig Venter thinks cryopreservation is old technology. He's not, and he's actually tried to stop my funding. He has done synthetic biology, which I'm sure you're very well of, and he believes, and this is probably an oversimplification, he believes that in the future we will be able to just put the codons in the computer and come up with that new life form, but unless you have a good library, you can't read it in. And so that's what cryopreservation does. it gives you the perfect book, and everything is intact. And since he asked this question, when you collect DNA, like say I want to collect Tasmanian devil DNA or something like that, and I collect it using a standard protocol, I actually break up that DNA. Many of the protocols that we have today causes that DNA to fracture, and cryopreservation doesn't. It keeps everything beautifully intact. So it's really a wonderful technology that is useful and cheap, and we know how to do it, and gives us that really important information.
But if you want to have to get the genome, you're going to have to perform the PCA to replicate the.
Right. You do break it up when you do that, but you do that known sites, and you can overlap. Okay. You had a question there?
Yeah, it's actually exactly this. Obviously, so you mentioned earlier in the presentation that obviously, yes, you hear about pandas. For example, I was in San Diego, I wanted to see pandas, they're rare to see.
Right.
As a dive instructor, what do you think is being done, or I'm curious as to how can we as people better influence the future generations to do this? I mean, is this something that goes back to dive organizations offering better education in colleges?
Yeah.
How do we get this so that it becomes, because like I said, I mean, I'm somebody who grew up as a recreational diver and changed my life as a beat diving instructor, and it's sad to hear because I can tell you, yeah, think about pandas, but you think about coral and you're like, oh yeah, it's damaging, but the presentation makes it very clear how important it is, whereas rainforests get this promotion, coral reefs don't get this information.
Right. And so the question she was asking is, how do we move from this spot where coral are not valued, really? And it comes down to values, really. And pandas are, which are genetic dead end. I just want you to know that they're genetic dead end. It has to, I mean, and some of it has to do with the cuddly factor, and we love cuddly things, they're furry, they're cute. But I think your point is well taken, that education is key in all of this, and how we value things, and how, I mean, I have to say, I think there's many things that change in our culture very quickly. I mean, in the United States, I never thought we would stop smoking, or promote not smoking. And it happened overnight. And when we had the disaster in the Gulf, a lot of energy, a lot of people were really focused on that disaster and what it was doing to the Gulf and our surrounding countries as well. I mean, we should not just depend on disasters, but I think your education really is the key, you know, so I don't really know I scratch my head all the time. Yes.
Is there a divers association, global association? So when you have an association, a lot of times what happens when it comes together?
I'm gonna get on what's his name? Here's Morgan.
There you go. You had a question.
Yeah, I want to react to that because we did a project with the small children today in the bay, and she came and talked to them and told about coral, and after that the children were so interested and they just went to circle around and look at all different things and brought all different kind of animals and species. And I think that's really the point. We need to educate the small children and let them look, and not just from a screen and
Right.
well, it's coral.
Yeah,
but
no.
let them experience.
Yeah.
And I saw a great difference.
Getting out is key in valuing it. Are we done? Thank you so much for your attention. I so appreciate it.
I got a big mouth. I can talk loud. Thank you very much, Mary. We'd also like to thank our sponsors tonight, Michael, Tricia, and the staff here at Brigadoon. What's coming up? We got lots coming up. Tomorrow afternoon, for those that have signed up for our hike with Adrian Del Neville, there has been a change. We are not hiking to Fort Bay. Adrian is going to do a hike from Spring Bay up to the Great Level, and you will have the opportunity to actually...
No, it's Spring Bay level.
Spring Bay Level. Who's doing the hike with Adrian? Do you know where to meet him?
Those are the things that I need.
There's lots of things that need to learn.